everybody. Welcome to another Expert Talk series. You know, we only spend about an hour and a half together, and that's very valuable. But your challenges are 24 hours a day, 7 days a week. We want to make sure that your needs are met as they come up. Need answers at 3 a.m. in the morning? Our free Everything ALS app has you covered. Download it from the App Store or the Google Play. We have our AI guide is always ready with answers sourced from our past expert talk. 24/7, you can find those answers. Plus, you can connect with our Pathfinders. And to me, that's one of the big advantages of the app is that there is a family inside. They're called the Pathfinders, and they've experienced things just like you. So if you want someone who understands what you're going through, open up that app, hit that connect button, and you will find the Pathfinders with our email addresses that you can contact us when you need us. We will be there. And with that, I'll turn that over to you now, Christian.
Thank you so much, McFinn. And it's my pleasure to welcome us yet again to another Expert Talk series. And today's guest is Dr. Shafeeq Ladha, who's going to be presenting Barriers to ALS Therapy Development, Forging a Path Through. And it's my pleasure to introduce Dr. Ladha. Dr. Ladha is the Chief of Neuromuscular Disorders and Medical Director of Muscular Dystrophy Center and the Director of the Gregory Fulton ALS and Neuromuscular Disease Center and Professor of Department of Neurology at the Barrow Neurological Institute. Dr. Ladha is the Ira and Mary Lou Fulton Chair in Motor Neuron Disease and has completed his neurology residency at the Barrow Neurological Institute with fellowships in neuromuscular disorders and EMG at the Mayo Clinic in Rochester and the Mayo Clinic in Scottsdale, respectively. He returned to Barrow, where he eventually founded the Gregory Fulton ALS Center, which has grown to be one of the largest ALS centers in the country. Dr. Ladha's research interests are in biomarker discovery and in clinical trial design. He has led numerous clinical trials in ALS and has over 60 publications in peer-reviewed journals. He currently serves on the NEALS Executive Committee, and we welcome him now. I will say a little bit of housekeeping before Dr. Ladha starts. As always, you are welcome to ask your questions in the chat. And at the end of Dr. Ladha's presentation, we will open it up to Q&A, and we'll make sure we get to every question. Dr. Ladha has been gracious enough to volunteer to stay as long as we need to. So with that, I'll turn it over to Dr. Ladha. Welcome.
Thank you very much. Thanks for everyone for attending. Thanks for inviting me. I'm excited to talk to you. Let me share my screen here. Can everyone see that?
Yes.
Okay, perfect. So I'm excited to talk to you today about therapy development in ALS, and you know, we haven't been that successful in that area, and a little bit of what I want to talk about is why have we not been that successful, and what can we do about it? Right? This is a really critical, important thing to understand if we're ever going to have better treatments or a cure for this disease. So see if I can get the just some disclosures. I do some consulting for companies that some of which deal with ALS. Here's kind of our objectives for today. So first, I want you to really understand what are the aspects of the disease itself that limit us in our clinical trial development and in our therapy development. Then a big part of this is going to be what are the solutions we're looking at. We've started to address these barriers with some solutions and haven't necessarily had a new successful therapy, but we have found ways that we're going to increase our chance of developing new therapies, and these solutions will go a long way to lead to that. And then finally, interspersed throughout the talk, we're going to talk about biomarkers a lot and kind of understanding some different roles of biomarkers. You know, it's a buzzword right now and an important topic, but there's a lot of ways that you can use biomarkers. And so I'm going to try to show you some of those and illustrate that for you. Okay, so here is the history of therapy development in a small little graph. And I put advances in quotes because many would argue we have not made dramatic advances in the way of therapy development. So back in the 1800s, Charcot, the French neurologist, described this disease probably in its first granular way of describing a group of people who had the characteristics of ALS. He's actually the one who named it amyotrophic lateral sclerosis. And then we went basically another 100 years before we had anything that made any difference with slowing the disease down. We got riluzole. We went another 20 some years and then got edaravone. And then after that, I took it off here because it was removed from the market. But obviously, we had Relivrio. But for sporadic ALS, we haven't made a lot of progress. Now, on the other hand, there's a ton of research. We've increased our amount of research in ALS dramatically over the last 20 or 30 years. So here's kind of a similar timeline, right? Starting back in the 20s, and these are number of publications per year. And you'll see kind of in 93, when Bob Brown described the SOD1 mutation, things started to really kind of take off and things got a lot more interesting for researchers. We had the SOD1 mouse, riluzole, the Niels Consortium, all of those things I think together sort of resulted in this dramatic spike. And now in the last 5 years, we've had, almost 3,000 publications a year on ALS. So, at some point that's got to translate to something, right? Well, what have we learned? Well, one important thing we've learned is about what causes ALS. And now we don't have a distinct single cause, but we do know a lot of the different biological pathways that lead to motor neuron death in ALS. And I'm not going to go through all of these. I think you guys in this group know a lot of them, but a big part of this is RNA and protein metabolism. So how are proteins made? How are they folded? How are they transported throughout the cell? That's a major factor that we've learned about probably in the last 10 or 15 years. We've learned a lot about neuroinflammation and these cells here called the microglia. We've learned about mitochondrial dysfunction, some of the stress granule dynamics. So there's a lot of things that contribute to this. And now what are we going to do about this, right? So a lot of our work has been elucidating this, but now we have all these targets, but we've been unsuccessful at drugging these targets to make important therapies. So why is that? Well, I think, to some degree, a fundamental concept that we have to question is ALS a disease or is it a syndrome? And traditionally in trials and in publications and in the clinic, we treat this as a disease, a single entity that one person has that is basically biologically the same as someone else who has the disease. And obviously over time, that seems less and less likely. First and foremost, the disease itself is very heterogeneous, right? You have people who progress quickly and slowly. You have people it starts in the bulbar muscles and others it starts in the arm. There's some people who have other aspects like cognitive aspects or Parkinsonism, or even sometimes sensory involvement. So all of that heterogeneity suggests to us that it seems likely that this may be more than one disease, or at least have many factors which contribute to what we see as the disease. And then when we look at more of the emerging science over the last 20 years, I think it becomes even more apparent that this is a syndrome with multiple pathways that lead to motor neuron death. This is a graph of all the genetic mutations that have been discovered since the initial SOD1 discovery. And each one of these genes can cause ALS, and they each have their own pathway that may be completely unrelated to other ALS pathways. So if you can have, for example, a dynactin mutation causing ALS, and dynactin, the gene actually codes for some of the proteins that allow for transport up and down the axons of motor neurons, and that's very different than SOD1, which actually codes for something that is that helps with oxidative stress. How how do we link those things directly if the disease is one disease? And so it suggests that multiple different pathways can cause the disease, and therefore this is likely to be a syndrome. And understanding that, I think, is at the heart of trying to decode the therapy development for this disease. So I'll give you some examples of this so it's a little bit more, I think, understandable. So here is a trial, let's say, that we're doing for this drug X. And let's say that drug X affects, protein misfolding, okay? Well, the problem is that some patients may not have protein misfolding. Maybe only these two green patients have protein misfolding as the major driver of their ALS. And now you do a trial, and you don't know who has protein misfolding, who has neuroinflammation, who has mitochondrial dysfunction. And lo and behold, the drug fails, right? The p-value is greater than 0.05. It doesn't work. Little do we know, in these two people, it worked really well, but their effect is washed out by all the people that were included who didn't have that mechanism, right? So this is a scenario where our poor understanding of who has what mechanism leads to a failed therapy that may actually work in the right patients. Here's another example, and this is one that I think is very common in our trials, and we've tried really hard to remedy this, but what about the impact of people who have slow progression, right? Another part of the heterogeneity of the disease. And you could imagine at the onset of the trial, it's hard to know who has slow progression and who has fast progression. But in this case, Arm A only has one person with slow progression. Arm B has a lot of people with slow progression. Obviously, irrespective of the mechanism of the drug, Arm B is going to look like it does better. If Arm B happens to be the placebo group, you may exclude a drug that actually works because your groups are unbalanced. And we don't have a great way right now of knowing definitively who's a slow progressor and a fast progressor. So these are just some examples of how the clinical features of the disease can really impact and be a barrier to our successful therapy development. So, as I've alluded to, we have this long history of failure in therapy development, and heterogeneity of the disease, slow progressors, fast progressors, different mechanisms. These are all big contributors to this long history of failure. But there's some other features. So our trials, I think, are too long and they're too expensive. When they're too long, it means people can't be in more than one trial. It means that one center puts in a lot of work to get one drug tested. The cost obviously is a problem. Anytime the costs are high, that's a problem. And you couple that with the fact that nowadays our pipeline is actually bigger than we can accommodate. We have the problem that's opposite of what we had 30 years ago, where we had very few drugs and lots of people wanting to be part of trials. Now we have lots of drugs in the pipeline that sort of overwhelms our patient population's ability to be part of these. So we need to really improve our trials if we're going to address these issues and stop this long history of failure. So a couple of ways we can do that. So certainly shorten trials. It's easy for me to say. I'll show you some examples of how we might do that. Improve drug throughput. Another thing that easy to say, but we're starting to make some headway into that. And accurately eliminate ineffective agents. So we want a process by which we can, upfront find a drug that doesn't work, and not only doesn't work, but doesn't work for really any of the people in the trial, and eliminate that so we don't waste any more time and effort on. But part of that is also not eliminating things that might still be effective in the right group. What are some things we can do? And this is not an exhaustive list, but I thought this was a useful set of examples of things that are being done now to try to improve clinical trials and therapy development so that we can have hopefully some success in the near future. So biomarkers are a big part of that. But in addition to biomarkers, we need better measures of the disease, and better measures in the human trials is important, but better models in the animal realm is important as well, and probably equally important. We'll review some of that. We'll talk a little bit about clinical trial design. What are some ways that we can change how we do these trials? Can we be a little bit more out of the box in our thinking to be more successful? And then a big one, I think, is improving diagnostic delay. All of us know that people with ALS go a year and a half before they ever get diagnosed after they've developed symptoms, and we're losing a lot during that period. And I think we can be much more successful if we can catch people a lot earlier. So we'll talk a little bit about that. Okay, so we're going to start with biomarkers. Now, before we talk about specific biomarkers, what are we looking for in a biomarker? It's a buzzword. Everyone knows the term, but what do we want it to do? Well, there's some practical concerns that I think are important, and then some scientific concerns. So the practical concern is it should be easily obtained, right? We don't want the biomarker to be a brain biopsy. That's not very useful to people living with a disease, let alone with ALS. We want it to be sensitive to a change in that disease. So as the disease progresses, the biomarker should proportionally progress, or at least progress in the same direction. And it should progress in a very small amount of clinical change. So if you lose a point or two on your ALS-FRS, we want the biomarker to capture that as well, and be sensitive enough to pick that up. We want the biomarker obviously to be related to the disease mechanism, and this is important from the standpoint of our therapy development. If we have a drug with a certain mechanism, we really want that biomarker to reflect that drug's effectiveness. And then we want it to change fairly quickly. Doesn't mean that the disease has to change quickly or that we have to have people who are progressing quickly, but the biomarker, if it goes down by 15% per month, even in someone who decreases by 1 point in their ALS-FRS per month, that's really powerful in that it allows us to detect a change quickly instead of over years. We can look at that over months. And that's an important factor for trials. And really in neuromuscular disease, not necessarily ALS, but a perfect example of this is a muscle enzyme called CK or CPK. And it's a simple blood test, so it's easily obtained. In people who have muscle diseases, it goes up when they get worse and it goes down when they get better. Obviously, it's directly related to a muscle disease. It comes directly from damaged muscles. And it changes pretty quickly. So if you give someone a treatment, it gets better within days. So it's a really nice biomarker for muscle disease. We want something like this for ALS. Now, how can we use these biomarkers? Well, first, an easy one is, can we use it for diagnosis? That would be great, because as of right now, we rely on the expertise of an ALS specialist to really make the diagnosis. And this can really improve diagnostic delay if we have a good diagnostic biomarker. We use it to track the disease over time. And we know that the ALS-FRS and the vital capacity and handheld dynamometry are reasonable measures, but there's a lot of noise in those measures. They kind of do this as they go. We want something that really can be tracked in a very objective numerical way that's very reliable without a lot of noise. We want it to be able to detect the effectiveness of a therapy. So can we have you give a drug and that biomarker moves in the direction of improvement. You stop the drug, it gets worse. That's something we want in ALS. And then, as I've sort of talked about earlier, we want to enrich a trial population. We want to basically find patients who are amenable to the mechanism of a specific drug and use the biomarker to choose them for the trial. I think that's really going to be a wave of the future. So let's look at some examples of these uses of biomarkers. Okay, first and foremost, here's just an example of some biomarkers that are out there. I'm not going to spend a ton of time on this, but you can see there's fluid-based ones and neurofilaments on here, but there's some others. There's imaging, there's neurophysiology like EMG and transcranial magnetic stimulation and things like that. And all of them have different roles and can capture different aspects of the disease and are being looked at in different studies. So those are kind of candidates. So all of you have heard of neurofilament. And, even though you've already heard of it, I do want to use it as an illustrative example of how it's being used and why it's being used. So it's probably these neurofilaments, there's multiple kinds. And right now, they're the most widely accepted biomarker. It's a fluid biomarker that can be obtained in blood and spinal fluid. And there's a few different types. There's neurofilament light chain, which is what I think most commonly people have gravitated towards. There's neurofilament heavy chain, there's phosphorylated neurofilament, and they all have slight differences. I won't really get into the advantages and disadvantages of each, but we'll more focus on neurofilament light chain because it's the common one. So these neurofilaments are present in neurons, and essentially when a neuron dies, it spills out this neurofilament into the surrounding fluids. And so we can measure it, and it's sort of a surrogate of how quickly motor neurons are dying. So that's sort of the theory behind why it was looked at. Now, it's important to understand we see this in ALS, but we see this in other diseases. We see it in Alzheimer's and spinal muscular atrophy, stroke, traumatic brain injury. But it's very clearly the highest in ALS. And so that's what makes it somewhat useful in ALS. So if you use it in the right setting, it can be helpful. If you just do it in every patient who comes through the door, you're going to get a lot of false positives. Now, it can potentially be very effective as a diagnostic biomarker. And here's some examples. On the left side of your screen, you'll see neurofilament levels in ALS patients, in patients who have diseases that mimic ALS, and then in healthy controls. And you'll see, clearly the highest, it's very high in the ALS patients. It's a little bit high in the disease controls. And then it's lower in healthy controls, which is what you'd expect. And if you generate a cutoff here, you can fairly reliably predict who has ALS just from their neurofilament level, at least in the hands of this group who's done that. The other thing that's important to understand is that neurofilament goes up before someone develops symptoms. So Michael Benatar's work in Miami has really documented this. Here is a group of patients who are carriers of, I think they're either all SOD1 or C9, and you'll see that their disease onset is at 0 here. And most of them, their neurofilament begins to go up before they get to symptom onset. And so neurofilament can not only help in diagnosing the disease when someone has symptoms, but they can also help predict who's going to get the disease. So very important and useful reasons to use it as a biomarker. It also can be helpful in prognosis. So here is a study that was done by the same group who did that study on the last slide. And you can see in green are the people who have the highest levels, and in blue are the people who have the lowest levels. And this is a Kaplan-Meier curve, which means it looks at survival. And you can see the people in blue actually had the best survival, and people in green with the highest levels had the worst survival. So this helps tell us that the higher your neurofilament, the sort of the more aggressive form of ALS you're likely to have. And this biomarker then can be very useful, and some people are using this now as an entry criteria to allow us to stratify people so that you don't get too many slow progressors and too many fast progressors in a given arm of a study. So I think we'll start to see this more and more in trials as something that's an inclusion criteria. Okay, what about other ways we can use neurofilament? Well, it's clearly it can be used as a treatment response, and we'll go into this in the next couple of slides. But it allows us in a very early type of trial, like a phase 1 or a phase 2 trial, where the primary goal is to look at safety or toxicity, and we can at least get a sense for, is this hitting a target that's important to us? If you do a safety trial and you see that patient's neurofilament goes down, that may influence a drug developer to say, okay, I'm really gung-ho about a phase 2 trial, versus if it doesn't move neurofilament at all, they may say, well, it looks like it's going to be safe, but I don't think it's going to work. And we may ignore that drug, throw it away, and start with something new. We've talked a little bit about this already, but neurofilament can then be incorporated for a variety of reasons now in trials. So we can determine phenoconversion in familial ALS. So this is what's being done in the ATLAS trial, where if your neurofilament goes up even before you have symptoms, you get started on tofersen immediately to see if we can prevent the disease from really kicking in or slow the disease down. As I mentioned, it can be used to stratify patients. It can be used to determine target engagement as a no-go decision. And potentially it can be used as a true surrogate biomarker. It has not reached that level yet with the FDA, but a true surrogate biomarker is a biomarker that does not actually look at the clinical effect and looks only at a biological effect and can be accepted by the FDA as a primary outcome measure for a trial. And we haven't reached that point, but the more we do with neurofilament, it could get to a point where moving neurofilament is enough to get a drug approved by itself. So that's a great segue. Speaking of using neurofilament in a trial, let's talk about tofersen. So we've talked about ways that we can use biomarkers and specifically neurofilament as a biomarker in clinical research. Here is an example of how that's actually happened. So tofersen, as you all know, is an ASO for people who have SOD1 ALS. And you can see on the left bottom panel is the neurofilament. In blue is the placebo group, and in red is the tofersen group. And you'll see neurofilament plummeted over the first 6 months of the trial. Dramatic effect on neurofilament. However, if you look on the right top panel, the ALS FRS score didn't really change that much between the two groups. They were pretty similar and not statistically different, which sort of led to initially the belief that this is a negative trial. However, when you actually followed this out further in the open label extension, we saw that the two groups, the people who got tofersen to begin with, after you follow them a full year, they actually did better than the people who started with placebo, showing that there was truly a benefit. It just took a little longer to be noticed. And I think if we had not seen this really dramatic improvement on neurofilament, number one, we may have missed this effect. We may have not been as aggressive about continuing the open label extension. And number two, I think very clearly the FDA would not have approved this drug. So this drug, although it did eventually show a clinical benefit in a post hoc open label analysis, we would have missed this drug if it had not been for using neurofilament. So a great example of where the biomarker showed us something that we would have missed otherwise. Okay, so we've talked about the first three of these. I'm going to touch on the fourth one, which is how can we use this to enrich a trial population. So we did this trial, Dr. Bob Bowser and I did this trial a couple of years ago, and what we did was we took an already FDA-approved drug called tocilizumab, which blocks a cytokine called IL-6. And IL-6 is a very pro-inflammatory cytokine. So what you can see here is sort of a mock-up of how we think neuroinflammation leads to motor neuron death. So you have a motor neuron in pink here. It dies for whatever reason. We may not know what the initial cause is, but when it dies, it spills out all of these misfolded proteins and mutant SOD1 protein and things like that, which then are broken up and stimulate these microglia, which are immune cells in the brain. The M2 microglia is a very nurturing, calming, neuroprotective form of microglia. And it exists in a back and forth state where it can become a M1 microglia, which is the opposite. It's pro-inflammatory, it's aggressive. And so upon encountering these misfolded proteins that are spilled from a motor neuron, the M2 microglia becomes an M1 microglia and then begins churning out IL-6 among other pro-inflammatory cytokines, which then drive this sort of toxic environment, damaging more motor neurons, which then spill out more of these proteins. And you can imagine a cyclical revving up of the immune system here to damage more and more motor neurons. So, our thought was IL-6 being so central, can we block this? Obviously, if we have someone whose major driver of disease is protein misfolding or mitochondrial dysfunction, blocking IL-6 is probably not going to help that much. So what we decided to do in this pilot study was, let's look at people who had evidence of high inflammation and only include them. So we took peripheral blood mononuclear cells, or PBMCs, from the blood of ALS patients. Before we ever entered them in the trial to get treated, they had their inflammatory gene profiles looked at. And we found that about half of the patients that we looked at had this high inflammatory signature with either really high levels of IL-6 or moderately high levels of IL-6 and increased levels of IL-8 and MMP-1, also pro-inflammatory cytokines. So we thought that this would be a good way to include people who had a high likelihood of responding to the drug. So here's kind of the way that the study went, and I don't have enough time to go through the whole study, but needless to say, we ended up rejecting about half the patients because they didn't meet that criteria, and only involving the high inflammation group. And we found that this was very safe. This was not a study that was powered for efficacy, but we found that it was safe. And some of the biomarkers we looked at, like C-reactive protein, which is an inflammatory biomarker, seemed to have a very, very dramatic response in the patients who were treated. We have not been able to get funding or drug company approval to do a phase 3 study, but this, certainly, I think, was a good proof of concept that we can do these biomarker-based enrichment studies with some success. Okay. I know I'm already going over, but I'm going to very quickly try to go through the last 8 to 10 slides. So we've talked a lot about biomarkers, and I think that's for good reason, that new biomarkers are going to be really important in all the ways that I kind of showed in helping us design and conduct better trials. But what about the actual trial measurements themselves? And most of you have probably heard how ALS-FRS is a good marker. It's not a great marker. There's some variability in it. Survival requires that we have very long trials, and so that's not very practical in this day and age. These traditional measures, they change slowly. There's some noise to them, right? On any given day, someone may have a point or two difference in their ALS-FRS just based on having a good day or bad day. That also makes them imprecise. And they may not always reflect response to treatment. So if the ALS-FRS kind of doesn't change a whole lot and may level off, it doesn't get better when we're successful with treatment. We'd like to see a marker that gets better. So we need better measures. One example is the Rhodes scale, and this is something that Christina Fournier at Emory has developed, and it's what's called a Rasch-Built scale, which just means that it's weighted based on some surveys that help tell us which factors are more important to people living with ALS. It involves a broader range. And so we think that this measure can be incorporated in trials, perhaps in the future, instead of the ALS-FRS, because it's more sensitive to change than the ALS-FRS probably, and can detect meaningful changes very easily. So this may be a measure that we can use. Home-based measures are really, I think, going to be used more in the future. And this really takes its power from the idea that any measurement has noise to it. And if you sample more frequently, you can average out the noise where you can see the actual signal that you're looking for. And so in the top right here is an example of that. If you just take one snapshot of this, this is just kind of an acoustic signal. And there's a lot of noise here. You can't really tell if there's any pattern there, right? But if you average it over and over and over again, now you see that there's these 3 peaks and they start to resolve themselves. And the same can be done if you do measurements more frequently with the vital capacity, or maybe using wearable devices, or even with the ALS FRS. So these frequent home-based measures could allow us to find a signal to show a drug's working in a much shorter time period and in a much shorter trial, therefore a less costly trial. What about models? We've talked about outcome measures in clinical trials, but we really need to, I think, improve upon our preclinical models. These animal models we use, they're very good if we want to look at mechanisms, if we want to understand the disease, but really they've been extremely poor at predicting a drug's success. And in fact, I would say almost that thus far, you could say 100% of the time they predict that a drug won't work, right? And that's just a sort of a tongue-in-cheek way of saying that every single drug that's ever shown a benefit in animals preclinically in sporadic ALS has failed in the phase 3 trial. So we need better models that give us a better chance of success. And one way to do this is cell-based. So these induced pluripotent stem cells are really, I think, going to be a great tool for us. Here you see all the different animals that we have, but now we need to add onto this armamentarium of some stem cells. And you can use these stem cells in a variety of ways. This is all thanks to Dr. Yamanaka, who won the Nobel Prize a while ago for being able to figure out you can take certain cells like skin cells or blood cells, you can expose them to the Yamanaka factors, which are these biological cytokines, and it makes these cells revert back to stem cells, pluripotent stem cells. And now you can take them and differentiate them. You can make motor neurons, you can make astrocytes, you can make microglia, and then you can put them all in a dish where it's a much more realistic system, right? Real brains in people living with ALS have all of these things that interact. It's not just a motor neuron problem. So we need these more realistic models to be able to develop better therapies. Here's just sort of how we might use them, right? So we take a patient, we make pluripotent stem cells, we create this brain in a dish that is unique to that particular patient, which is really nice, which now allows us to either model the disease, so we can do different things to those cells and see how they behave. We can use the cells as a cell therapy, so we could give these cells back to someone as a stem cell kind of therapy, or we can use them to screen drugs. We can put, these cells in 1,000 dishes and use 1,000 different drugs and see which drug helps the motor neurons live longer. And then that allows us to find that candidate drug that we can take to trials. Clive Svensson has done some really interesting work in California looking at not just making these brains in a dish, but making a sort of a blood-brain barrier with a blood side to it. So this is an even more realistic model where you have the brain side, you have the blood side, and you have a blood-brain barrier, and you can actually put sort of synthetic blood through there, and you can measure what's happening with the brain side, looking at neurophysiology, all kinds of different things, which allows you to, create different models to test drugs and test drug delivery, even all kinds of things. So this is really something that I think will allow us to be doing a much more personalized medicine approach. We can take someone's individual cells, make the brain in a dish, or make the blood-brain barrier, brain-blood chip, and test different drugs and see if they might work before we start putting them into patients or an individual patient. What about clinical trial design? How can we improve on that? I've already alluded to, this is just illustrating the lifespan of a trial is very long. It's 2 to 4 years, and it's very costly. The more time and staff you need, the harder it is. So as most of you know, the Healy platform trial has really tried to address a lot of these shortcomings. So we can now have a clinical trial infrastructure that gets reused instead of having to kind of close it down and restart all over again. It's the idea that instead of, building a stadium, having the Super Bowl and tearing it down, you build the stadium and now you have the Super Bowl and you have the World Cup and you have all of these things. you have concerts in between, and you have simultaneous use of this infrastructure that allows us to more quickly get through drugs and more quickly get us a go, no-go decision. And it's been very successful in that way. The Healy platform trial, this is kind of a little bit of an old slide. We now have regimen F and G that have gone through. So in just a couple of years, we've gone through, 6 or 7 different drugs. Unfortunately, none of them have been a slam dunk effective drug, but we've certainly been able to rule out some drugs and sort of get them out of the pipeline very quickly. And then there's other drugs like the nanocrystalline gold and predopidine, which may go on still to clinical trials based on this data. So this is, I think, has been an example of ways we think of clinical trial design. I didn't have enough time to talk about some of the other things that we're using, like machine learning to create, virtual controls for a trial, or modeling a trial before we do the trial to look at inclusion and exclusion criteria and see which ones might be most effective. These are all techniques that we're using now to make trials better and faster, and more efficient. Okay, and then in the last couple of minutes, let's talk about diagnostic delay. This is, as I've talked about, a very big problem. Why is this delay an issue? Well, first of all, we're getting people in trials when they've lost a lot of their motor neurons. Is this too late for these drugs to work? We'd prefer, I think, in trials to preserve motor neurons rather than try to have to replace them. And right now, the only approach that we have would be really real success would require replacement because people have lost so much. I wonder, and I think there's some examples of this with the transcranial magnetic stimulation data that's out there. Does ALS have different biological phases? So, on one hand, is there a sort of a beginning triggering phase that kind of converts into almost a prion-like automatic cascade where it's on autopilot later on? And do we then need 2 different kinds of approaches to a disease that may have different gears to it, so to speak? And then finally, I think, do we set ourselves up for failure? Do we end up testing drugs that are effective if we were to test it very early, but not effective late? It would not surprise me if that's happened to us in certain drugs. In terms of how can we shorten the diagnostic delay, I think just awareness is an important one for neurologists. And I think there's been these tools and campaigns out there to help clinicians. The motor neuron disease red flags tool in Europe, the ThinkALS tool here. I like to use this in my lectures to residents and med students and primary care doctors. When you think, you should think of ALS when you see someone with painless, asymmetric, progressive weakness without sensory symptoms. That's a little tiny blurb that really captures, not a lot of things do that. So if you're a neck surgeon or a hand surgeon and you hear that, you see this, you should think maybe ALS and maybe not a pinched nerve. And that, I think, will hopefully help shorten that diagnostic delay. Biomarkers, I think, will help us with this too. Neurofilament may be a really good one if we can get people to test for it early. Testing, these other biomarkers still are limited by the fact that someone has to consider ALS as a diagnosis before they order these. So biomarkers are still going to be limited in their ability to shorten this diagnostic dilemma. Okay, last slide. What are our future approaches in therapy development? Well, I think we've talked about so many mechanisms. I think that means that we need to be drugging multiple mechanisms at the same time. So are we going to have cocktail therapy, multi-drug therapy? And I think it makes a lot of sense, and that's in our future. Personalized medicine is going to be very important now that we can create these cells in people and hopefully start to figure out what mechanisms drive the disease in individual people that will allow us to find drugs that work specifically for a given individual. finding people pre-symptomatically, especially in the genetic forms, is going to be really impactful. And then, what's the future of stem cells? I know there's a lot of excitement about it. I personally feel like we may not, we may be overselling it currently, and we may not have the tools yet to make stem cells as powerful as they could be for therapy development. But I think in the future we'll build on that. And then collaboration is critical. It sounds obvious, but the ALS community has done a fantastic job of this. Other communities that I interact with, and I won't give any specific subspecialties. I don't want to insult anyone, but other subspecialties in neurology can be very competitive and people don't share, and those fields tend to stagnate. So I think if we can keep this collaboration going, it's going to be really powerful. All right, well, let's hope. All of this I've talked about, the hope is that it leads to more advances and we can take away the quotations and have real advances. And over the next 20 years, begin to have, instead of just a small treatment effect, begin to have a very substantial treatment effect, or better yet, a cure. And I thank you for your attention, and I'm happy to take some questions.
Thank you, Dr. Ladha. And we do indeed have some questions for you. So I'll start off. Anush is going to join me here as we go through some of the questions that get us going. The only biomarker, but this is from one of the audience members, the only biomarker I've heard of is neurofilament. Are there any others that are in the works or that you would say are as invested in or kind of on the pipeline like neurofilament was?
Yeah, I think neurofilament is clearly ahead of all other biomarkers and has been used pretty extensively at this point in trials, whether they be phase 2 or phase 3. There are a lot of other biomarkers that are being used, but many of them are a little bit more specific. So for instance, I use the example in the talk of neuroinflammation and using the gene expression profiles. So that is, considered a biomarker, but people may be using, SOD1 protein is another good biomarker, but it only applies to SOD1 patients. So I think there's a lot more biomarkers and they're being used in different ways. Not every biomarker can be used for every role. Some might be diagnostic, some might help us in tracking the disease over time, but not both, right? There are, neurophysiologic markers that are being looked at, like transcranial magnetic stimulation, things like that. So there are a ton out there, and they're being used in trials as we speak as supportive measures for those trials.
All right, so the next question is, you talked about the role of neuroinflammation. So are people who are not high inflammatory the slow progressors?
Well, I'm not sure we actually know that yet. So the first question is, what's the very best way to measure neuroinflammation? And I gave you an example of that in this presentation, but I'm not certain that that's the ideal way to measure neuroinflammation. Inflammation is extremely complex. Something goes up, another thing goes down. It can change over hours or days. So depending on when you measure it may make a difference. So I think the field hasn't sort of agreed upon an ideal measurement of neuroinflammation, which then makes it very difficult to use it to now predict who's going to be slow progressors or fast progressors. I think we may find that it is, I think the field believes that it is a modifier or a modulator of disease progression. So I don't think neuroinflammation causes ALS, but if you have a lot of neuroinflammation, you probably progress faster. If you have less, you may progress slower. But again, we don't have the tools yet to definitively show that.
Thank you. Do you have any knowledge of the phase 2 Fossadil trial? There's some interest in the results.
I do not. I'm not privy to those results.
Thanks.
Next question is, the Healy trial protocol is now longer and excludes anyone over 24 months post-clinical symptoms. So what kinds of efforts are being made in the field to kind of find ways of including people at scale for these platforms?
Yeah, so this was a big, discussion among the platform trial organizers. And, this actually came from a lot of statistical and machine learning modeling showing that, if we change the criteria around, what are the chances that we're going to get a group that's going to give us a successful trial? I won't get into how exactly that decision got made, but it did lead to a little bit more restriction in that. And I think that, we've seen a lot more expanded access programs over the last couple of years. Certainly through the ALS Act, that's helped, but there's other ways to get involved in expanded access programs. And I think we have to be, as a community, really focused on providing those opportunities so that people who can't be part of the clinical trial directly can be part of the expanded access programs. And I think most of the sort of the big names in drug development recognize that, and most of us are working with companies to try to bring those to our clinics.
Thank you. Question from the audience is, my neurologist has speculated that Radicava may only be effective in the very early stages of the disease. I've also heard some say that Relyvrio may be effective for certain patients. Are there any studies designed to test these hypotheses specifically?
I wish. I think it would be fabulous. The practical problem is that more trials takes money, and money has to come from somewhere. And the companies that make these drugs have, in general, decided they don't want to do more studies. Either it's not worth it to them, or they don't have the money. In the case of Amylyx, so these are challenges. It takes a lot to do these trials, and we don't have great large funding sources that are easily accessible. You can make the same argument about a lot of the drugs that have been tested that have failed. Did we just not pick the right people? If we could go back and redo the trial and change how we did it and maybe select different patients, could we have had better success? And so I think there's drugs out there we missed. And how do we get those back into trials? And that's a tough question without funding.
Thank you.
The next question is, will the FDA recognize that treatments in mouse models don't translate to humans and stop requiring animal studies prior to clinical trials?
Yeah, I think they have in large part. I think most of the time the FDA does not require that you show the drug works in an animal model, but they do require animal model tests, or at least animal testing, to look at toxicity. And I think they would like to see that you use an animal model to hit your target, or that there's a substantial biological demonstration that your drug is doing something that we want it to do. So I think there's still a role for these animal models, but I think the FDA recognizes this limitation.
Got it. Thank you.
Thank you. And the next question is, how is NIL, I believe it's NFL collection, done? Should we all get it tested?
It can now be done as a simple blood test. It's also done as a CSF marker. There's been some comparisons between the two, and I think generally now we think that the blood-based marker is almost as good as the CSF, to the point that it's probably not worth getting CSF clinically. In research, we still do that. Should we all be tested? I think this depends on clinic to clinic and what you're doing with it. From a research standpoint, we use it for very different reasons than we use it in the clinic. I tend to get it on my patients when they're diagnosed, mostly because it gives us a little bit of a prognostic feel for how they're going to do. If they have a very high neurofilament level, I might expect that people are going to progress a little faster than if they have a low or a very low neurofilament level. I think clinics are different in how they do it. I think you just want to talk with your neurologist and say, do you think I need neurofilament, and what would we do with that? We don't want to just send you for blood just to get, blood work done and just to do it. We want to have a reason.
Has there been any studies that look at slow progressors versus fast progressors and seeing if there's a difference in neurofilament light?
There has, certainly. We know, I showed a slide earlier that the people with lower neurofilament have a more likelihood of having slower progression. The people with higher neurofilament at the beginning tend to have a faster disease progression. So, there is, now the problem is, you all know the disease is heterogeneous. So once in a while you see someone who's a very fast progressor and their neurofilament's low, or you see someone who's really slow and they have a high neurofilament. So it's not a perfect test. And it's great when you have an average group and can make predictions, but that prediction in a group of people doesn't necessarily always translate to an individual person. And so that's the limitation of using it in the clinic.
I'll jump in also in that, here at Everything ALS, we're really focused on developing digital endpoints and actually digital biomarkers that can help increase the precision and subpopulation definition of people with ALS. Do you see value in pairing neurofilament specifically with these functional efforts, like digital speech measures to strengthen readouts?
Yeah, I think, this is where AI comes in. And again, AI is another buzzword. Everyone thinks AI can do anything, but the place that it really, I think, can be powerful in ALS is by taking huge data sets with multiple different parameters and looking at it in a million different ways faster than any human can ever look at it, and identifying a pattern that may be useful. So in your example, Christian, if you had a bunch of digital biomarkers, neurofilament levels, forced vital capacity, ALS-FRS, and you had all of them in a huge data set of people, it might be that AI says, oh, look, the people who have, greater than 45 degrees movement in their arm for the first 6 months and a neurofilament level that's below this level and a forced vital capacity that drops by 1% per month, those are the people that do the best, right? And we would never figure that out by just looking at raw data as a human. So I think we're going to have these kind of multimodal predictive models where we have multiple different markers that we look at all at once to form those predictions. And we kind of already have that, right? So the TriCal's predictive modeling looks at a variety of different things, and that's kind of how they came up with that, is using machine learning. But I think that's a very early entry into how we would use these many different kinds of biomarkers to look at.
Thank you.
Are there any other diseases that have similar high heterogeneity to ALS, either in progression or in patient population, where the research community has kind of found strategies to overcome some of the trial barriers that you were talking about, and that we could potentially learn from or apply to ALS?
Yeah, that's a great question. I'm not sure I have a great answer to that, but maybe a different way to answer that question is, I think cancer used to be like that, right? So let's say you're in the 1700s and there's a lump in your liver or a lump in your lung. I think, at that time they just said that this is cancer, right? And it would get treated all the same, and probably at that time there were probably no good treatments. And then the microscope came about, and all of a sudden we could look at it and say, wait a minute, these cells in this cancer are different than those cells. These are two different diseases. And so I do think that we're in the pre-microscope era with ALS, and we need that microscope to be able to get to the heart of that heterogeneity. And so cancer, I guess I would say, yes, cancer has been through that, but it was, generations ago. I can't think of anything off the top of my head right now. There probably is an example currently that I don't know about, but I think that illustrates the same point.
And I'm going to follow up with that. As you talk about cancer, one of the big things in cancer is the amount of education that's been done effectively and really bridge the gaps between community cancer and specialty centers. You mentioned education efforts to general neurologists that have been done. Could we be using the development of these AI kind of analyses to accelerate or simplify the path to being able to diagnose or at least characterize high likelihood or high risk people prior to or alongside actually developing true biomarkers?
Yeah, I mean, I think we can. I will say that, first of all, the ALS neurologists or the neuromuscular specialists generally don't need any other tools. Someone has ALS by seeing them in person, and that's all you need. So who do we target, right? General neurologists, other kinds of physicians, maybe people who we would target. Where would the AI come in? Would it be in their clinic? Would it review records? And so the limitation of the AI in ALS is that it doesn't know good data from bad data. And by data, I mean, I'll give you an example. I saw a patient who they actually used an AI medical tool, and it told them they thought they had ALS. And so they came to see me, and they very obviously didn't have ALS at all. And they had basically a really poorly done EMG test. And the AI didn't know what a good test and a bad test was. And so it took the data and just interpreted it very, upfront and gave this person the scare of their life, right? So somehow that aspect of ALS has to be taken care of. And I think that's a challenge because, there's a lot of testing that goes into ALS, which means there's a lot of false positives or false negatives. And all of that is just going to get spit out by the AI without really a good way of looking and saying, is this a false positive or not?
Thank you. We just got a question in the chat as well. That's actually a couple of layers to it. So do you think, first of all, that we need a paradigm shift in the way research is conducted? With hundreds of aberrant cellular processes involved, it is unrealistic that one or even three substances will fix everything. It would be a nightmare to analyze an elaborate protocol that attacks everything at once, but the goal should be to solve the problem first and figure out which substance is correcting each problem later. Just about everyone involved works for someone. Are they beholden to the ones who sign their paychecks to find a profitable solution, or what's the issue?
Yeah, I mean, I think that first of all, we do need a paradigm shift, and I think everyone knows the paradigm shift that needs to happen. We just simply don't have the tools yet to make that paradigm shift, and that is that we need to be able to see a patient, take blood, put it through a battery of biomarkers, and say, you have ALS type A, or you have ALS type B, or type C, or type D, et cetera, and on and on. And then say, if you have type A, this drug works on that pathway. Let's get you into that treatment, right? That's the shift that will make all the difference in the world and give us some real success. We just don't have the biomarker panel yet that's reliable, that can tell us that. So I think that's what we need. I don't think that we're holding on to a paradigm that is ineffective because of business. And I know sometimes it can feel that way. I certainly will tell you that any of my colleagues and myself, if I got put out of business because there's a cure for ALS, I am all for it. That's all I want for my patients living with ALS. And I don't really, even the drug companies, most of them I find really deep down have good intentions in getting real treatments. And so I think they would embrace a new paradigm if we had the tools to make that new paradigm happen.
All right, the next question is, what is the correlation with autoimmune diseases in ALS? And if you have an autoimmune disease, are you more likely to develop ALS?
Yeah, so we don't think that having an autoimmune disease makes you more likely to develop ALS. There's not any real evidence to demonstrably show that. And I'm not sure, autoimmune is not exactly the issue here. It's an immune response. So we call it neuroinflammation, which is that diagram I showed you. The immune system gets revved up by all of these abnormal proteins that it's not used to seeing. And just that inflammation then just damages nearby cells. So that's a little bit different. And so again, I see that as a driver of disease progression. Inflammation doesn't cause the first motor neurons to die and release those substances. It just picks up those substances and now contributes to amplifying what is already happening in motor neuron cells.
Thank you. I think that we're getting close to the end here, but we'll have one more question. Oh, sorry, just another question came in, so I'll go to that one first. Can AI be used to build a virtual ALS brain where drugs and combinations of drugs could be tried in this virtual brain to see the effect?
Yeah, I mean, I think that would be a wonderful way to use it. I'm not sure I'm not sure our society
Yeah.
would trust that as of yet. We've got a ways to go to where the AI has to demonstrate that approach can be successful, because it's a big leap to just show that a drug works in this AI model and then just put it into people, right? I think most of us would want a little bit more, but that may be something in the future that is used in ALS research.
Thank you. Thank you. Dr. Ladha, we're over time, but we do have a couple more questions that we'll send your way. But thank you so much for the time. You've been incredibly generous with us to stay over. Really appreciate all the explanations you've provided. And of course, if anybody has additional questions, feel free to continue to email us and reach out to the Pathfinders as well. We work regularly with them as they are constantly in the field with you, our people in the community, to answer any of your questions. So Dr. Ladha, once again, thank you. And I'm going to turn it back to you, McFinn.
Well, Doc, I want to ask you a personal question.
Sure.
What did you feel about the questions that came your way tonight and the people who attended? Is this typical for you?
Yeah, I mean, as I kind of, before we started the webinar, I mentioned to you that I'm always impressed when I give talks to people living with ALS about how sophisticated the questions are and how much understanding they have for these topics. So it's I think par for the course for me, but certainly it's always impressive every time I give a talk, and it's what makes it fun. I enjoy questions that are challenging and demonstrate understanding of these topics.
Well, we just want to make sure that you're taking care of yourself personally.
I try to.
Okay. Okay. Well, see, we depend on you. And we don't really want you to stay up late, but we really do. And so that's why I say, you better be taking care of yourself, because there's a lot of people who really appreciate what you do, what you say, and who you are. So,
Well, thank you.
all of us, you are our hero today. Thank you, sir.
Thank you for having me. Everyone here is a hero to me for living through all of this. This is, I can say I understand, but obviously I don't really understand. I can understand as much as someone who doesn't have ALS can understand, but I applaud everyone for their perseverance with this disease and people's great attitudes in the face of such, terrible bad luck.
Dr. Ladha, we can live through this because we have individuals like you who are fighting for us. Thank you.
It's my pleasure. It's my great honor to be part of this work.
Well, folks, all I can say is the doc came for us. We got to come for him. If you ever want something from a large group of people who have lots of opinions, we would love to share our opinions with you.
All right. That sounds great. I may take you up on that.
Okay. That's what we want.
All right.
Called teamwork.
Yes. Wonderful. Thank you, everyone. Have a great night.
All right. Thank you so much.
Thank you, Doctor.